Hello, this is our third session of evolution, and I'm repeating the slide on heterozygote advantage because there is a certain concept that I want to deliver today. In heterozygote advantage, meaning that when you are hybrid, then this simply means that you have a certain advantage in your population. I'm sorry, I have to make sure that I am sharing the proper screen. So what is the advantage and what heterozygous individual are we talking about? We are giving the example of sickle cell anemia. And last time we said that if you are heterozygous dominant, if you are homozygous dominant, S capital, then all your hemoglobin in the blood is normal. So you have this privilege of being normal, of not suffering from sickle cell anemia, yet you are sensitive to malaria. So in areas where malaria is endemic, like in Central Africa, for example, we will find that those individuals that are normal for the sickle cell allele die from malaria. Of course, if you are homozygous recessive, then you will die early in life because this means that both alleles are recessive in your genotype and that most of your hemoglobin is liable to sickling and that all your red blood corpuscles will be hemolyzed and destroyed, or you might die not from hemolysis and severe anemia, but you might die from clotting and loss of function of a particular organ if the artery to that organ is clogged by the clot. Our example of malaria and sickle cell anemia focuses on the heterozygote individuals. As you know, the S small allele or the lower S represents a recessive allele that is responsible for abnormal hemoglobin. If you are heterozygote, this means that 10 to 20% of your hemoglobin is liable to sickling. And you might be wondering how, if the presence of the dominant allele is supposed to silence the recessive one. But the fact is, this is a condition of incomplete dominance. It's totally different from complete dominance, where the dominant allele totally masks the recessive one. It's also different from a lack of dominance, when both alleles appear on the phenotype and their effects blend. It's also different from co-dominance, when the two alleles are expressed and they do not blend, like in the MN blood group. Here, this is a condition of incomplete dominance, when the recessive allele can be partly expressed in the presence of the dominant allele, but under certain conditions. So under certain conditions, the dominant allele is no more completely silencing the recessive allele. So for example, you will find that 10 to 20% of their hemoglobin is abnormal, and the symptoms of anemia will appear only if they are exposed to certain conditions like hypoxia at high altitudes, for example. The presence of those heterozygote individuals that are resistant to malaria is very interesting, because being resistant to malaria, then if they are present in Central Africa, they will have advantage over the other two genotypes, over the homozygous dominant that are totally normal and the homozygous recessive that show the sickle cell anemia. But having only the sickle cell trait makes you resistant to malaria, therefore being able to reproduce, therefore being able to pass on your alleles to the offspring. And this is why, and this is a very important concept, this is the one I was targeting by repeating the slide, that the mutation of the sickle cell trait will be preserved in the gene pool. Maybe when we talked about heterozygous, the heterozygous state in evolution before, we said that the presence of the recessive allele masked or silenced by the dominant allele leads to preservation. This is very important, that's why I'm repeating it. It leads to preservation of the recessive allele in the community, and maybe one day that recessive allele would be or would prove to be advantageous under certain conditions, and this is what occurred in the case of sickle cell trait and malaria. In frequency dependent, notice, sorry, Notice that we are discussing the factors that help to preserve variation in a population, because natural selection is based mainly on the variation that occurs due to mutation. Mutation is the raw material of variation, and natural selection helps make use of this raw material of variation to help on the long run development of new species. So there are factors, there are endogenous factors in every population that help preserve the variation. And one of the most interesting ones is the frequency dependent selection. In the world of animals, when animals search for a prey, they have a certain search image for the prey, meaning that they have to know, they have to have imprinted in their memories what prey they are looking for. So usually this search image is that of the more abundant and common phenotypes, meaning that the animal would not be looking for a rare animal to be considered a prey. Otherwise, the animal wouldn't find anything to eat. So what happens exactly that the minority here is an advantage. If there is a species of animals that is abundant, then it would be targeted for preying, for being a prey, I mean, prey with E. But if you are a minority, then most of the time you will be ignored because the search image in the brain of the predator, the animal that wants the prey, is the common phenotype. But the problem is, so again, let's repeat this. We call this a minority advantage. So what happens actually is, being minority from the beginning, a minority from the beginning, you are safe. You will not be preyed upon. And those common phenotypes will be preyed upon. But upon preying them, their numbers will decrease. And they will decrease to an extent that they will be no more present, almost extincted. So the animals, the predators, will shift to the minority, to the animals that were previously a minority, and they will start preying on them. Why? Because during the time when the predators were preying on the more common phenotypes, the minority was living a very easy life. They were reproducing, they were increasing in number, and they no more became a minority. So when the previous majority was extincted, now the minority will be preyed upon. So frequency-dependent selection preserves variation in a population. How? Because it acts to decrease the frequency of the more common phenotypes, and it increases the frequency of the less common ones. How? Because during the time when the common phenotypes were preyed upon, the minority individuals were reproducing at ease without the presence of any competition. So as I told you, in predator-prey relationships, the predators develop a search image. Or what is a search image? What is the definition of a search image? It is the standard representation of the prey that enables them to be focused on a particular kind of prey. If the prey individuals are different, meaning that here by different, we mean that we have variations, so they are variable. There are several variants. Then first, the most common types will be preyed upon disproportionately, meaning that we'll be focusing only on the most common types, while the less common individuals will be preyed upon to a lesser extent. And here comes the minority advantage. We are being preyed upon less extensively. This is the minority advantage. But it is to be noticed that the minority advantage is temporary. Why is the minority advantage temporary? Because you didn't notice, you, the minority, that while you are reproducing happily, you are being changed into the majority, and therefore from now on you will be considered the most favorite prey. So since these rare individuals, and hereby rare individuals, we mean the minority, temporarily have, as I told you, this selective advantage. Selective advantage of what? Selective advantage of being a minority. They will become more common for a time, and they will lose this selective advantage, and you will be selected against. Why eventually are they selected against? They are selected against simply because they are preyed upon. And why are they preyed upon? Why are they being selected against? Because they are turning to be the majority. They are turning to be the majority. So it's a very interesting example. And in biology, we have a famous example, which is the one of the hare and the lynx. The lynx, of course, is the predator, and the hare is the prey. And the graph in green represents the number of animals in thousands, and this represents the years. You can see that the intervals here are intervals of 10 years. So what happens at the beginning? For example, let's take this this part of the curve where the hare is increasing in number. Why are the hare increasing in number? Because the lynx is decreasing. And why did the lynx decrease? Because in a previous period, the number of the lynx were increasing just because they were feeding on the rabbits. So every time we have, say, for example, that the let's explain it in a simpler way. Say, for example, that we have 10,000 hares, and then we have only 1,000 lynx, the lynx, the animal. And say, for example, that those 1,000 had their search image the hare. So what happens is that they will totally devour those 10,000 hares. And what happens that, so they will increase in number, and those ones will decrease in number. But after a while, what happens is that they no more find hares to prey on and to prey upon, and therefore they will die out of hunger. So the lynx will decrease again in number. And during that time where the lynx decrease in number, the animals or the prey, the hares, will increase in number. So every time you will find a crisscross between the graphs. For example, here you have the lynx that is increasing. Maybe here it would be a better and easier representation. Here you have the lynx that is increasing just because it is eating the hare that is decreasing. So most of the time you will find, of course, the two graphs are not exactly synchronized as I explained, but in many periods of time you will find that as the hare as the lynx is increasing, or when the lynx is increasing, this means that they have found enough hares to feed upon, and that's why the number of the hares is decreasing. And then finally, we will talk about the evolutionary neutral traits. What do we mean by an evolutionary neutral trait? We have learned that a trait that you possess, but we cannot talk about the trait absolutely, but we have to talk about a certain trait in the context of a particular environment. So if you have a specific trait, but we have to consider it in the context of a specific environment, then this trait might give you a selective advantage or a selective disadvantage, meaning that this trait might be favorable or unfavorable. But again, it's not absolutely favorable for all types of environments, and not absolutely unfavorable or disadvantageous for all types of environments. You have to read the effect of the trait on your on the reproduction of the animal in the context of the environment. When we talk about evolutionary neutral traits, we mean traits that are not known to be causing a certain selective advantage nor a certain selective disadvantage. Of course, I think that this piece of information is not very up to date because I think it has been changed, and I read several articles against this. For example, so again, let's see the definition of an evolutionary neutral trait. There are traits that seem to have no selective advantage. Actually, not only no selective advantage, nor disadvantage, because they are neutral. And they gave the example of the blood types in humans. And they say, when we say this means that having a certain blood type like O or A or AB cannot give you a certain advantage, meaning that the scientists have not found contexts or situations where a certain blood group may represent a selective advantage or a disadvantage. But actually, this is not correct, because there are recent articles that talk about certain blood groups that are correlated with a certain increased resistance to diseases or decreased resistance to diseases. There are other scientists who have another hypothesis. They say that they are not, of course, okay with the previous opinion, and they say that how come they do not have a selective advantage? We think that those blood groups that have persisted in populations up till now, like blood group O, blood group B, blood group AB, and blood group O, are the ones that were selected for. And those scientists, on the other hand, have another hypothesis that there were more than these alleles we know. We know the allele A, we know the allele B, and we know the allele O. But their hypothesis says, or they hypothesize that there were more alleles, but unfortunately, the individuals with those genotypes, including those alleles, did not survive, and that only those that persisted because they were selected for. And this counteracts the first hypothesis, or contrasts the first hypothesis that says that they are evolutionary neutral traits. Up till now, there are studies to see whether the presence of a certain blood group affects survival, for example, affects reproductive success, and therefore continuity of the species or not. So again, we have the neutral traits, and this leads us to the definition of a neutral mutation. And a neutral mutation is a change in the DNA sequence that is neither beneficial nor disadvantageous or nor detrimental to the survival and the reproduction of an organism. We always talk about this, survival and reproduction. By this, we would have finished the first part of evolution. I now stop sharing, and I will start sharing the second part of the presentation, which is evolution part 2. Okay, evolution part 2. Okay. Now, why is it stopped? Okay, then let's make sure we are recording. Yes, we are. And we are, right? Yes. And because I have many bad experiences about spending hours and hours thinking that I'm recording and then finally discovering that I'm not, or I am sharing the incorrect screen. Now in evolution part 2, inshallah تطلع okay. Then we are discussing I don't know what's happening. Okay. We will be discussing interesting concepts, like, for example, the factors that cause evolution. This is exactly the opposite of what we were discussing at the beginning, but they are not contradictory, meaning that in the previous part one of evolution, we were discussing the factors that preserve variation in the population, factors that preserve variation in the population. And now we are going to see how we will make use of that variation, or the evolution will make use of that variation. So what's the paradox here? No, the paradox is not here. I'm sorry. The paradox is at the end, when we are going to study something very interesting about the stability of the populations, which is the Hardy Weinberg principle. So let's start by the agents that cause evolution in a population. We have number 1, genetic drift, number 2, genetic flow, number 3, mutations, number 4, non-random mating, and number 5, natural selection. These are 5 important factors that we have to memorize because they are important factors that cause evolution. Number 1, genetic drift. Number 2, gene flow, mutations, random mating, and natural selection. Okay. I see a small window on and it doesn't want to disappear. Okay. Okay, what is a genetic drift? The genetic drift describes like random changes that occur in the number of the alleles, of the various alleles in the population that occurs by chance, meaning that it is not prepared for, it is not planned for. These variations in the number of alleles, we call them changes in allele frequencies or changes in the percentage of the alleles. As we said, genetic drift is I'll stop sharing and share again because this window is very troublesome. It hides most of the, again. Okay. So genetic drift describes, as we said before, random changes in allele frequencies in a certain population, or random fluctuations. Fluctuations means changes, but rather increase and decrease, increase and decrease. And as we said, they are random, meaning that they occur by chance. They are not planned for, they are not prepared for, and they are unpredictable. So when you define genetic drift, you have to talk about a change in allele frequency. You have to talk about fluctuations in the numbers of gene variants, and the gene variants are nothing but the alleles. But you have to remember the adjectives that you can use. They are random fluctuations. They occur by chance. And as I said, fluctuation means that there is an increase followed by a decrease, and they are unpredictable. Okay. In order for the genetic drift or such fluctuations in allele frequencies to be seen, we have to have a small population, because in large populations, what would happen? In large populations, there is what we call dilution of alleles, meaning that if there is a change, that change will be diluted by the big number of individuals in the population. Genetic drift, as we will see, will limit diversity. It will limit diversity, it will limit variation, but it will result in the development of new species. And we have 2 examples that are very interesting. The first one is the bottleneck effect, and the second one is the founder effect. The bottleneck effect talks about natural disasters, like fires in a place, earthquakes and floods. So actually, what happens is that there are many people who pass away during these catastrophes or those disasters, and this will result in the reduction, of course, of the size of the population because many people died. And the reduction is not selective, meaning that there is no selection. I'll not select, for example, people who are white-skinned or people who are dark-skinned or people who are tall or people who are short. You can never know which disaster will kill which individual. So since it reduces the size of the population, the number of the individuals will be smaller. But at the same time, it will not be representative of the original one, meaning that if this is the gene pool of the population, and that is an interesting term to learn, the gene pool of the population means all the alleles that can be present in all the individuals. So in the gene pool, you have a representation. When you think about a gene pool, then, for example, this gene and that gene and that gene. So we have all kinds of genes that we can represent, for example, by different shapes. So this is the gene population, all the alleles. It's not, of course, it's not a pool, it's not a container, it's something abstract. We talk about a gene pool, about all the alleles that might be present in a population. It's more logical to think that if there is an earthquake, for example, that destroys buildings in a particular place, then death should be at random, unselectively. So why would it favor a particular group of alleles or individuals? This is what we're going to explain now. So the fact that the resulting population is smaller is logical because there are people who passed away. But why is it not representative of the original population if it is random, if it is unselective? Why does it result in a shift of genetic variation, meaning that there is a change in allele frequency that will occur? So in the bottleneck effect, we're talking again about genetic drift. So it is an example of genetic drift. There will be a genetic drift, meaning that there will be a genetic shift. And what does it mean that we will have a genetic shift? This means that some alleles will be underrepresented, and other alleles will be overrepresented. Now, is it meant? Is it on purpose? Is it planned for? No. This diagram is amazing for you to visualize or to understand what the bottleneck effect is. Imagine that this is the original population containing those beads of all colors, purple beads, orange beads, green beads. And if you have a look at the original population, you can see that there is no, maybe it was the orange one that was taking over. So we might say that in the original population, the orange is taking over. And then there is a catastrophe that occurs. What is this catastrophe? We call it a bottleneck event. Why is it a bottleneck event? Because it kills most individuals. Okay, they stay in the bottom, they cannot descend. Maybe because those individuals here, because the beads are bigger, sorry, they are bigger than the mouth of the bottle, and only those are the survivors. And because they are the survivors, then they will reproduce, they will pass on their traits and increase in number. And this is the new population. You can easily detect that in the new population that arose from this minority that survived is totally different, because now it's the purple phenotype, for example, that is overriding or that is taking over. When we shaked the bottle, we shaked it unselectively. but it happened that those that survived, that descended in the bottle, were having more purple, and therefore we have a new population. The examples of the bottleneck effect, there are two famous examples. The first one is the Tay-Sachs disease. There is a high frequency of Tay-Sachs disease among Eastern European Jews. Why? Because in the Middle Ages, during that period, many Jews were persecuted and killed, and the population of Jews was reduced to a very small fraction of its original size. And among the survivors, it happened totally randomly, totally unselectively, totally by chance. There was a disproportionate percentage of people who carried the allele for the Tay-Sachs disease. And the Tay-Sachs disease is a disease of the neurons that is characterized by deposition of certain chemicals in the brain, and those chemicals were, one second, those chemicals were called gangliosides, and those gangliosides result in impairment. When they deposit, they are not supposed to accumulate in the brain. When they accumulate in the brain, they affect the function of the brain in the newborns and may result in mental disability. The problem is that Jews in Europe, because of all that happened, remain isolated, do not intermarry with other people, so they do not outbreed. They interbreed, and therefore Tay-Sachs disease remained high in the population. This is a famous example of the bottleneck effect, and as you see here, Tay-Sachs disease is a rare disease. It is a genetic inheritable disease characterized by problems in the neurons, leading to death in the neurons of the central nervous system. That makes it very serious. It's not in the peripheral nerves, but it is in the brain and in the spinal cord because of accumulation of gangliosides. And the other example of a bottleneck effect is the northern elephant seal. Of course, the northern elephant seal, we don't have a picture of it here, but it was hunted to extinction, meaning that they hunted it extensively in California from the 1820s to 1880s, like a 60-year period. And then in 1884, they set laws to put this elephant seal under governmental protection, and it was banned to hunt it. And therefore, the remainders, the survivors of the elephant seal started reproducing again and increasing in number. But it happened that all the descendants had very little genetic variation, because maybe when the hunters were hunting the northern elephant seal, as we said, they hunted it to almost extinction, meaning that maybe there were only two parents, a male and a female, that stayed alive, and therefore there wasn't adequate variation. This would not be like when you start with thousands of individuals to make a population. Now, the founder effect is another example of genetic drift, but here we don't have catastrophic events, we don't have natural disasters, but the founder effect differs from the bottleneck effect in that it's all related to migration. So, for example, we have a small population that breaks away from a larger population and they go to colonize a new area. And of course, because it is a small population, then it is not genetically representative of the original larger population. And here, by the original larger population, of course, what matters is the gene pool or the genetic pool of that original population. So here, in that small population that broke away, you will not find all the alleles that belong to the original genetic pool. And therefore, there could be some rare alleles that might be overrepresented. rare alleles should not be overrepresented. Rare alleles should stay rare, but because it happened that this small population that broke from the original population and migrated to other areas, like, for example, the famous example of the Old Order Amish of Lancaster in Pennsylvania, where it happened that a small group of settlers came to the United States from Germany in the 1770s, and they had a dominant trait, but that dominant trait was very rare, which is the allele that is expressed to cause polydactyly. What is polydactyly? Polydactyly is having a, for example, on the hands, you will have a sixth finger like this, and similarly in the feet. So you have extra fingers and toes, and most of the time, this extra finger is short and it may be adherent to the finger before. Of course, what made the founder effect be like intensified in that population is, just like we explained in the case of Jews, those people, the settlers, also showed isolation and intermarriage of the close community. For example, intermarriage between cousins, first cousins. And that's why there was a high incidence of polydactyly. So in the founder effect, it's not that there was destruction of the whole population and only a small representative stayed alive, but here there is a small community that migrated and went to a new area and started increasing in number. And accidentally, as you see, the small population that decided to migrate had this dominant allele overrepresented, and therefore when they reproduced, especially that they are isolated, especially that they are they interbreed, so it was overrepresented. So the factors here were, you started by a small population that was not. So the factors that helped in this condition the founder effect to occur is that you started by a small population, and in that small population, some alleles were overrepresented, and then there was migration. But what helped this is the isolation that occurred, and number three, the interbreeding or the intermarriage in case of human beings. You might be asking, in the previous example, you might be asking yourself, how come that the gene or the allele, I'm sorry, for polydactyly is dominant and at the same time rare? This seems paradoxical because, as you know, a dominant allele usually silences a recessive allele. Okay? So whether a trait, as you know, is dominant or recessive, whether an allele is dominant or recessive, it will determine if it is supposed to be expressed or supposed to be hidden. But at the end of the day, you cannot guess how common the trait will be in the population because of many factors. So again, if I have a dominant allele, say, for example, P capital for normal fingers, P capital for polydactyly, P capital for polydactyly, and P small for normal fingers, normal count or numbers of the fingers. So that of polydactyly should silence the normal one. And therefore, since that allele for polydactyly is dominant, why aren't all people having polydactyly? First of all, the allele of polydactyly, in case of genetic drift, for example, it was overrepresented. It was overrepresented because of the founder effect. That doesn't seem weird because this is not the problem. The problem is that the opposite. How can a dominant allele be underexpressed in a population? Why? Because it under normal conditions. So we knew why in genetic drift it was overrepresented. But the other important factor is, yes, you are a dominant allele. Okay, and you should be proud of being a dominant allele. But I'm sorry, are you useful to the individual or non-useful to the individual? If you are advantageous, then you will be selected for. If you are disadvantageous, then you will be selected against. And that what happens in for polydactyly under normal conditions. If we don't have a founder effect, if we don't have a small population that migrates and that accidentally had a bigger concentration or frequency of alleles, and that rarely occurs, the normal is that the allele for polydactyly, although being dominant, will not show on the majority of the population because it is disadvantageous. For example, let me give you one simple example. And here comes the role of the environment and the role of epigenetics. So it's not all about being dominant or recessive. It's about the environment. Are you favorable to the environment or unfavorable to the environment? If you are coping with the needs of the environment and the needs of the individual, then you will be selected for and you will flourish. And the opposite occurs if you are not okay. So let me give you one example. Why don't we see many people with polydactyly despite having this dominant allele? Just maybe because people don't want to marry somebody who has polydactyly, and therefore they don't find a mate, and therefore they don't reproduce, and they don't pass on this trait to offspring. The second factor that helps the occurrence of evolution, of course, if you are asking yourself what's the relation between genetic drift and evolution, in both cases, whether in the case of Tay-Sachs disease or in the case of polydactyly, we had two species that were, or two small groups of populations that were isolated, that developed into a new species where polydactyly is predominant, or where actually we cannot say a species here, but we can say new variants, a new population where it's not a species because they are still human beings, I'm sorry, but there is a new population that is different from the original population and that shows a higher percentage of polydactyly or a higher percentage of Tay-Sachs disease. The second factor that might help in evolution and the development of populations with new characteristics is the movement of alleles into a population, which we call immigration, or because of immigration, or the movement out of a population that we call emigration. Okay, of course, we needn't the whole individual to be to migrate. And of course, it has to be a fertile individual. If it is infertile, it is not in our concern. But we might have migration or flow of gametes. So usually, gene flow is the transfer of individuals with their gene pool inside, or transfer of gametes. But you needn't have the whole individual, just like pollen grains, for example. Pollen grains may be carried by air to distant areas. This is one example of gene flow. And of course, it helps increase diversity because you know that in flowers, for example, there is self-pollination. So if this is the ovary of the flower, pollen grains from one flower may fall on the stigma of the same flower. So this diminishes diversity. But if the pollen grain flows and migrates and goes to another area, it might pollinate another flower, and therefore this would increase diversity. Mutation, of course, helps evolution because mutation is a permanent chemical change in DNA, in the genetic material. And as we said, it is the raw material for evolutionary change, because variation initiates diversity or creates diversity. Now, just a single, see how mutations are important. Just a single point mutation that is not corrected might result in a new allele into the population, and this is the case in sickle cell anemia. We all know that sickle cell anemia is an example of a single base mutation. It's a substitution, actually. You might be asking yourself, but mutations are rare. For one locus, if you consider one locus, how frequent can a mutation occur in a particular locus on the chromosome or in a particular part of the DNA molecule? They're supposed to be very infrequent. Maybe for the whole year, you might have 5,000 mutations. But the problem is not with the rate of mutations per locus, but it is in the cumulative effects of mutations if they are not corrected. The fourth factor that might help in preserving evolution or allowing evolution to occur is non-random mating. Usually, individuals select mates for specific reasons. As we saw, for example, the females select males with the most conspicuous plumage, the brightest plumage, because these males are the ones that are capable of protecting them. This is, if you select your mate for a specific reason, then this is called non-random mating. For example, if we say that human beings or human males, select females who are of average height. So all females that are too short or all females that are too tall would not find a proper husband and would stay single and unmarried. So this means what on the long run? If this is repeated, actually, alhamdulillah, this does not occur in human beings. But if it occurs, then the less fit individuals will be automatically eliminated from the population. Why would they be eliminated from the population? Because they do not mate. وبالتالي, they do not have offspring. They do not have viable offspring, and therefore they cannot pass on their traits to other individuals. For example, snow geese. Snow geese are of two phenotypes, white and blue. Usually, white geese tend to mate with white geese, and blue geese tend to mate with blue ones. But if for a certain reason, the blue geese became more attractive, and both the blue and the white geese would start searching for a mate that has blue feathers, then on the long run, the population would change rapidly, would evolve rapidly. So this would be an exceptionally rapid type of evolution. Why would it be like this? Why did white geese change their preference? Maybe because there were environmental stresses. It's not normal that the white geese change their preferences all of a sudden, unless you have an environmental stress that makes their environment more risky to them, and therefore the white geese start looking for the blue geese that are more attractive, and not only more attractive but more powerful and more capable of protecting them. In number 5, we have natural selection, and natural selection is, of course, the major mechanism that causes evolution. But again, natural selection cannot work unless there is an original variation. So the population has to be variable first. We should start by having variation, and then after that, natural selection would act on that variation. So as we said before several times, there are certain individuals who are better adapted for a particular environment. They are, as we said before, they are not the best, but they are the best fit individuals. So they will reproduce more, they will exhibit better reproductive success, and they will have more offspring, they will survive, They will survive first. We should have written survive first, and then having reproductive success and more offspring, and can pass on their genes to their offspring. I'm sorry, I got a bit distracted because it's raining heavily outside. And now we're going to talk about an extremely interesting example. It's not only extremely interesting, but it is extremely important, and it is frequently encountered on the tests. And this is what I talked about at the beginning of the presentation, that talks about exactly the opposite of what we started the presentation with. We are not now talking about an evolving population, but we are talking about a stable population. And that principle is called the Hardy Weinberg principle, or Hardy Weinberg equilibrium. And it talks about the characteristics of a stable population. And by stable population, we mean a non-evolving population in which the allele frequency does not change. So in all our previous examples, when we were talking about evolution, we were talking about gradual cumulative changes in allele frequencies in the population, which is the definition of evolution. But here we are talking about the characteristics of a stable population. For example, if the frequency of a certain allele of a particular trait in a population is 0.5, 0.5, by the way, means what? 0.5 means 5 over 10. It also means 50 over 100. It also means 50%. So that allele represents 50% of the alleles for that trait in the population. Say, for example, there is a dominant allele for a particular trait and a recessive allele for a particular trait, and the recessive or dominant allele represent 50%, then in 1,000 years, it will stay as it is if there is no evolution. So there are certain characteristics for a population to be said. I'll pause for a while because it's getting too noisy from the rain, and then I will resume recording. Hello again. So there are certain conditions for a population to be considered a stable population, or for a condition to for a population to achieve that condition of being stable. First of all, we cannot talk about a stable population where we can see the Hardy Weinberg principle and apply the equations of Hardy Weinberg if the population is small, so the population should be large. Why? Because even when a small change will occur in that population, in the frequency of alleles or in the gene pool, from now on, when we say a change in the gene pool, we mean a change in the allele frequency, then this will be diluted by the sheer number of individuals. We have a huge number of individuals. So even if a change occurs, that change will be diluted. And therefore, one of the major conditions is the population being large, in order to make sure that there will be no change in the frequency of the alleles, and therefore if there is no change in the frequency of alleles, the population would be stable. On the other hand, if you have a small population, then the smallest change in the gene pool will have a major effect in allele frequency, because it would represent a significant, I would rather say, instead of a major effect, I would rather say a significant effect in allele frequency. Okay. Now, what other characteristics do we have for a stable population? Of course, there are very difficult conditions to be achieved. For example, the population must be isolated from other populations, because if it is isolated, then you can make sure that there is no migration of individuals from the population, nor would there be like immigration of individuals into the population. This would help stability of the population. There should be no mutations occurring in the population. Of course, these seem to be impossible conditions because, of course, a mutation will cause a change in allele frequency. And you might be asking yourself, if the conditions are that impossible, why is the Hardy-Weinberg equilibrium and equation so famous? Just because they help us calculate frequencies of alleles in an evolving population. So when Hardy-Weinberg, when they set this equilibrium or the conditions for an equilibrated stable population, they didn't mean that there is a population like this in the world, but they just set the conditions for a stable population so that if you are not a stable population, if there is an evolving population, we can do our calculations based on that equation. Also, mating must be random because we said several times before, if we have selective, random here means non-selective. Because if we are selecting only those individuals that are well adapted and that have the reproductive advantage, of course, the other options will disappear. So mating must be random to keep the population stable, because if mating becomes selective, then there would definitely be a change in allele frequency. And of course, there should be no natural selections, because natural selection does not diminish the number of individuals, does not diminish the number of variants, as we said before. But here there is a very important description that you have to memorize. It changes the relative frequencies of alleles in the gene pool, meaning that if I have 3 alleles, A, B, C, for example, allele A had a percentage of 30, allele B had a percentage of 50, and allele C had a frequency or percentage of 20. Then, for example, if allele C is the most adapted to the environment, then natural selection will make on the long run that the frequency of allele C would be 60%, for example, that of allele B 20, and that of allele A 20. So what happened actually, that we did not diminish the number of variants, we did not diminish the alleles. They are still 3 alleles and they are the same, A, B, and C. But what changed is not only their frequency, but their relative frequency, meaning in relation to each other. So what's the importance of the Hardy-Weinberg equation? First of all, it enables us, am I recording? Yes. It enables us to calculate, as I told you, the frequency of alleles in a population, and therefore you can deduce the change. And of course, we are going to cover certain simple examples, but it can be applied to rather more complex situations. And it can also be applied to very simple cases when we have 2 alleles, for example, a capital and a small. Now, there are certain rules for being able to use the Hardy-Weinberg equation properly. So P small will stand for the frequency of the dominant allele. So, for example, if in a population, the frequency of the dominant allele is 60%, then the frequency of the recessive allele for that trait, when we talk about relative frequencies of 2 alleles, we talk about alleles that control the same trait. So this 60% will be denoted P, whereas the 40% will be denoted Q. So again, P stands for the frequency of the dominant allele, and Q stands for that of the recessive allele. How did they get this equation? They started, actually, I should have written this first. It's true that both are equivalent because, as you know, in algebra, if you have A plus B equals 1, then if you square both sides of the equation, then A plus B squared equals 1 squared. And therefore, you can expand this, and you can have So when you expand it, it will be A squared plus 2AB plus B squared equals 1. So the idea is that if you have, as we said before, the percentage of the dominant allele denoted to P, and if the dominant allele is 0.6, 0.6, as we said before, means 60%. So it is a frequency, because you might be asking yourself, what's 0.6? How can this be the frequency? Yes, because 0.6 is 6 over 10. It is 60 over 100, and therefore it is 60%. So definitely, if for a particular trait, the dominant allele has the frequency of 0.6, then the recessive allele would have a frequency of 0.4. So it all starts here. And then we square both types of the equation, and we have P squared plus 2PQ plus Q squared equals 1. And it is essentially related to the monohybrid cross. Why to the monohybrid cross? Because if you have a monohybrid cross, if you have two individuals, PQ, and now we are not using an uppercase letter and a small case letter, but we're using two different letters in order to be able to use the same formula as Hardy Weinberg. So we should write them both lowercase. Okay, so if I have a hybrid, if I want to represent the dihybrid cross, the monohybrid cross, the monohybrid cross, so I'm crossing two individuals, both being hybrid for one individual. So I will have PQ, this is the first, this is dominant, this is recessive, this is the first parent, the genotype of the first parent, and this is the genotype of the second parent. And then you do the gametes, okay, P and Q. And then here you have the gametes, again, P and Q, and you do a Punnett square. And of course, you know the results would be what? So you would have P, this is the homozygous dominant individual, this is 1. And then you have 2 P, Q, and then you have 1 Q, Q. What does this mean? This one is that one, which is, is that one, P with P, okay? And then we have 2 that are P with Q, and then 1 that is Q with Q. So we have one individual that is PP, we have 2 individuals that are PQ, and we have 1 individual that is QQ. This is the ratio, the ratio you have here, which is 1 to 2 to 1, is the ratio of the monohybrid cross. This PP, we call it in the equation, is corresponding to P2, and we will say why. So P to the power 2 means the individual that is PP, that is homozygous dominant, and Q square means that individual that has both recessive alleles. So here, P squared means an individual whose genotype contains the 2 dominant alleles, and Q square means the individual whose genotype contains both recessive alleles. And the heterozygous individual that has 1 dominant and 1 recessive allele is denoted PQ. And now there remains an important point, but it's a bit outside the scope of the curriculum. Why, if my homozygous dominant individual has the 2 dominant alleles, we represent it by P squared. P squared means P multiplied by P. Why? Because if you are made from an allele P and another, a gamete P and another gamete P, then the probability that you have P, let's call P the probability that you have this allele in your somatic cells, and this one, the other allele, the probability that you have the other allele in your somatic cell. And since both are independent and non-linked, then the probability of having them together is the multiplication rule, or P multiplied by P. This is not important. Nobody will ask you about the proof but this is why we write it in the form of p squared. Maybe when we see the example, you will be more acquainted, or you would assimilate more what I said. Now, in a population, we have 9% of the individuals that are blue-eyed. And here there is a very important thing to know in order to avoid mistakes. If I say that P is the frequency of the dominant allele, then the frequency or the probability of the homozygous dominant individual will be P multiplied by P, or P squared. So P squared is the frequency of the dominant population. Frequency of dominant population. I mean the individuals that are homozygous P or homozygous for the dominant allele. So again, this is extremely important. So I have to translate the words of the problem, the word problem, into one of these formulae. So 9%, first of all, means 0.9, or can be written as 0.9. This is more convenient because we have P plus Q equals 1. So if you use a decimal, that would be more convenient. Blue eyes, this is recessive. So, and this is a percentage in the population. And this is a recessive gene. So 0.9 represents what? First of all, will I use the letter Q or P? I will use the letter Q because I'm talking about a recessive individual. But are we giving you that percentage? Does this percentage we are giving you represent the frequency of an allele or the frequency of a population? It represents the frequency of a population, and therefore we can call it Q squared. We can call it Q squared. Okay? So 0.9, now we have translated the first sentence into a small equation. So what percentage of the population, again, is hybrid for brown eyes? Then we are asking about Q squared, because so this is what is given, and this is what is required. Okay, so let's first solve A. So the trait for blue eyes is homozygous recessive, okay, and therefore we deduced that Q squared, because we are talking about population, is 9%. So, sorry, 9% is not 0.9, 9% is 0.09. If I say 0.9, this is, I'm so sorry. If I say 0.9, then this is 9 over 10. It's good that I made this mistake in order for you to avoid it. So 0.9 equals 9 over 10. As you know, this decimal equals 9 over 10, which is 90 over 100. But if you want 90 over 100, of course, is 90%. But if you want to formulate 9%, then it has to be 9 over 100. Then it has to be 0.09. So Q squared is 9%. Then if we convert it into a decimal, it would be 0.09. Therefore, from this, you can deduce that Q equals 0.3. Why are we reverting it to Q? Because the initial equation is P plus Q equals 1. So if Q equals 0.3, then P would be 0.7, which is 70% of the population having brown eyes. Now, notice that they are asking about the population that is hybrid for brown eyes, because you might be asking yourself, if Q is 0.3 and P equals 0.7, how much of the population are blue-eyed and how much of the population are brown-eyed? But the brown-eyed individuals are sometimes pure and sometimes hybrid. In the question, they are asking about hybrid individuals. How do we represent hybrid individuals in Hardy Weinberg principle? To PQ. We already have the P, we already have the Q. The P is 0.7, we calculated it, and Q is 0.3, we calculated it. So the percentage of the population that are hybrid for brown eyes is 42%. And those, of course, that are homozygous homozygous dominant is represented by p to the power 2, meaning 0.7 to the power 2, meaning 49%. Another example for Hardy Weinberg principle is determine the percentage of the population that is, again, we are asking about not because in Hardy Weinberg problems, you have two types of questions. Either we ask you about the frequency of an allele, or we ask you about frequency of population. When we say frequency, we mean percentage. So determine the percentage, again, of the population. So they want it in the form of a percentage, and they want the percentage of the population that is homozygous dominant. How can we represent this? First, we have to ask ourselves, how can we represent it? We can represent it by p squared. P because it's dominant, squared because it is homozygous. So this is what is required. What is p squared? And what they give us is the percentage of the population, again, that is homozygous and recessive. So this is the percentage of homozygous recessive. How can we translate those two words or formulate them into the symbols that are used in the Hardy-Weinberg equation? So a homozygous recessive individual, I'm not talking about the frequency of the recessive allele, but I'm talking about the homozygous recessive population whose percentage is q squared. So we can formulate it as q squared. So q squared equals 0.16, meaning that q equals 0.4. And then we are going to make use of that. Since q equals 0.4, then p equals 0.6. Why? Because p plus q equals 1. Now that we got p equals 0.6, then we have to get p squared, which would be 0.6 to the power 2, or 0.36, or 36%. This was the second example of Hardy Weinberg. And now here are some important points to remember about the part that we covered in evolution. Number one, evolution occurs at the level of the population, meaning that no single organism will change in response to a change in the environment. We mean because in evolution, what do we have in evolution? In evolution, actually, we have a change not in the phenotype. This is important because you might be asking yourself, how come that a single organism does not change in response to a change in the environment? We are talking about the frequency of alleles. We are not talking about phenotypes. We are not talking about phenotypes because, of course, as you know, the phenotype might change because of the environment. I might be tanned because of the sun, or more complexly speaking, we can talk about epigenetics and the effect on the environment, but the effect on the environment is actually an effect on gene expression. It is not an effect on the genetic code itself. In the evolution, you do not change the alleles. You do not initiate a mutation. The mutation is initiated spontaneously, and the evolution works on that mutation. So the final result of an evolution is a change in allelic frequency, a change in allele frequency in a population, and that change is, so it has to be at the level of a population, as we said before, and it has to be a gradual cumulative change. It has to be a gradual cumulative change. I'm not sure about the M. I have to look up this. So it is the frequency of the allele that changes, and we denote the frequency of the allele by P or Q, but the percentage in the population is denoted by P squared or Q squared. Here we have a third. I'm trying to get you all the examples of the Hardy-Weinberg principle. Here we're going to determine, again, the percentage of a population. So it is either P squared or Q squared that is hybrid, So it's neither p squared nor q squared, because the percentage of the population that is hybrid is 2pq. So whenever you're asked about the percentage of the population, if the population is, let me write it here, if the population is homozygous dominant, the population in question is homozygous dominant, then what we are looking for is p squared. If the population in question is homozygous recessive, then we are looking for q squared. And if the population in question is heterozygous, then we are looking for 2pq. This is what is required. Now, what is given? What is given is the allele frequency, not the population frequency of the recessive trait. How can we translate this? We can translate it as Q. Okay, so here we are not given the value of Q squared, but we are given the value of Q. So all what you need is to write this simple equation, P plus Q equals 1, and since Q equals 0.5, then P will be equal to 0.5. And since both P and Q are equal to 0.5, then it's easy to calculate what is required, which is the 2pq. So we are going to multiply 2 by 0.5 and then by 0.5 again, and we get 50%. This is the percentage of hybrid individuals. And now we're going to talk about speciation and reproductive isolation, but after having paused for a while. Hello again. We're going now to talk about speciation, or the formation of species, and how it is related to reproductive isolation. Before introducing to the concept of reproductive isolation, we should know first what a species is. A species is a population, a group of individuals whose members can interbreed, as we said before. I think we mentioned the definition before. They should be capable of interbreeding, not only of interbreeding, but of getting offspring, and those offspring have to be characterized by being viable and being fertile. For example, in the example of the horse and the donkey that we gave before, we had the mule, and the mule was alive, was viable, it's capable of surviving, but it is not fertile. And if it is not fertile, then this individual will be the last individual because it will not give more offsprings. Some individuals, as we said for the horse and the donkey, another example, lions and tigers, for example, they can be induced to interbreed when they are imprisoned, in captivity, means that they are not in a forest, they are not in a wide land, for example, where they can meet other lions or other tigers. But in nature, they wouldn't do so. So if lions and tigers interbreed, then they interbreed because they are forced to do so in captivity. But if they are free, they wouldn't. And therefore, the lion and the tiger, although they can interbreed, they are considered to be a separate species. So what, again, we are going to continue our talking about a species. The second characteristic of a species is that it is manifested by reproductive isolation, meaning that one group of genes will become totally isolated from the other group of gene, thus preventing interbreeding. And therefore, each of the two groups of individuals whose alleles are separated will have a totally separate evolutionary history. For example, if two animals originally belonged to the same species, but one of them lives in water, for example, and the other lives on a high mountain, and it is extremely difficult, say, for example, that it is an animal that creeps, for example, it would be difficult for that animal to reach the top of the hill. Therefore, the gene pool for that group of individuals and the gene pool for that group of individuals are totally isolated because the two individuals cannot breed with each other, they cannot mate. And every one of these two groups of individuals or populations will be affected by different environmental stresses, by different environmental selective pressures. So every one of these two groups will be exposed to different environmental selective pressures, and therefore, on the long run, they might end up by being two different species. So again, they started as two subpopulations or two subgroups of the same population. One of them, because it was more adapted to that aqueous environment or that watery environment, lived in water. The other one, although belonging to the same species, got adapted and got lodged in that niche. The niche is the environment that is most favorable to you. The niche, you can say that the niche is your bubble. So if an organism or if a population selects to live in a particular environment and finds it favorable, we consider that environment the niche or the comfortable environment of this organism. And because they are wide apart, despite belonging to the same species, but why are they wide apart? Because they are variable. Yes, they are variable. They are not similar to each other, and everyone got lodged in its niche. So there is no reproduction between them, and this is what we call reproductive isolation. And at the end of the day, once separated, the two isolated populations may start to diverge genetically, meaning that at the beginning, what do we mean by diverge genetically? At the beginning, they belonged to the same species, which we consider here the same ancestor, for example. And then there was some like geographic separation between population A and population B. Let's call this one population A and population B that belong to the same species. The longer the duration of time, the more the selective pressures will act on each of the two groups. How can the selective pressures cause this divergence? Because the selective pressures will work on the gene pool. They will be favorizing some alleles and unfavorizing other alleles. And the same applies to population B. But of course, the alleles that were favorized or unfavorized are different from those of population A. And at the end of the day, they diverge totally because of the selective forces that affected them. It's exactly similar to, or almost similar to, when you have, for example, even a sibling who has long been living in Europe and you're living, for example, in Egypt. At the end of the day, when you meet, you will find differences. This we cannot call evolution. I'm sorry that I said exactly similar, but I'm just talking about how those two different environments even affected your phenotypes, the way you behave. Of course, they didn't affect your alleles. They didn't change your alleles. They didn't change your relative, your alleles. And there is no population to say that there is a change in the relative allele frequency, but there was a change. We cannot call this change evolution, because evolution has to be mentioned in the context of a population, but we can just call it a change. So in order for a new species, because I've been asked during the last class, why are you saying a species if they started by being two populations that were reproductively isolated from each other? So because they will end up by time, or as time passes, they will end up by being affected by the selective forces favorizing and unfavorizing alleles. So we need time and we need selective forces. So reproductive isolation here was very important, because the gene pool on the top of the mountain was totally isolated from that at the bottom, and they couldn't mate. So speciation is anything that will fragment a population, that will isolate small groups of individuals, and at the end of the day, they will end up by being different species. When we say, so we start by we start by a population and we end up by having a species. There are different modes of speciation. We have allopatric speciation, the one we talked about when there is separation by geographic factors like mountains, like rivers, like whatever geographical barrier that might separate the two subpopulations. And we have sympatric speciation. Sympatric speciation is speciation or fragmentation of a population to prepare for the development of a new species on the long run. We call it sympatric speciation. There are 5 important factors that help sympatric speciation: polyploidy, habitat isolation, behavioral isolation, temporal isolation, and reproductive isolation. Polyploidy, as you know, it's when you have more than 2 complete sets of chromosomes, more than 2n. So you might have 3n, 4n, 8n, but we usually talk about 4n and 8n because they are the commonest. And as you know, polyploidy might result from non-disjunction during gamete formation, during meiosis. So if it happens that we have non-disjunction during gamete formation, then one gamete might be 2n instead of being 1n, and the other gamete will have 0 chromosomes. So if it happens that during sperm formation, the same occurred, and we have a 2n sperm, notice that a 2n egg is totally abnormal, and a 2n sperm is totally abnormal. Is if they fertilize each other, we would have a 4n individual. Similarly, if this 2n egg is fertilized by a normal, here the sperm was abnormal because it is 2n, and this one, of course, that contains 0 chromosomes will be abnormal. This egg is also abnormal. So abnormal with abnormal will give 4n, and 4n plus 4n would give 8n. But if an abnormal egg is fertilized by a normal sperm, then at the end of the day, we would get an individual that is triploid. Of course, if you have a plant that is polyploid, because polyploidy is very common in plants, they cannot breed with other plants of the same species that are not polyploid. Why? The reason, this is not the time to mention the reason because we would spend a lot of time, but actually they do not breed with each other. So a polyploidal plant can interbreed with a polyploidal plant. A polyploidal plant cannot interbreed with a non-polyploidal plant. So actually, at the end of the day, there would be like separation between the 2 populations because they cannot breed. But here, they cannot breed not because there is a geographic barrier between them, but because there is a functional barrier between them, one being polyploidal and the other being euploidal or having the normal number of chromosomes. Similarly, we have in sympatric speciation, habitat isolation. In habitat isolation, there is no geographical barriers like huge mountains or hills, for example, or rivers between the 2 populations, but they live in the same area. There is no geographic isolation, but they rarely encounter each other, and therefore there is no opportunity for them to mate. Like, for example, there are 2 species of one genus, because you know the genus is broader than the species. So if you say genus, this is larger, and then you have the species, okay? And then you have at the very bottom of the chain, you will have the population. So if they are in the same geographic area, but one lives in the water and the other one is terrestrial. Of course, the terrestrial one is very close to the one in the water, but actually, why would the terrestrial one bother itself descending in the water. It wouldn't be adapted to it. So the one in the water reproduces with that in the water, and the one on land reproduces with the ones on land. You cannot consider this a geographic barrier, but actually there is reproductive isolation. So all the examples we are going to see, whether they are those of allopatric geographic speciation or non-geographic sympatric speciation, are examples of isolation, impossibility of reproduction, and that's why the big title was speciation and reproductive isolation. Because whatever the type of speciation is, it is based mainly on impossibility of reproduction because of reproductive isolation. So if we go back, that reproductive isolation can be geographic reproductive isolation if we talk about allopatric speciation, or it can just be a functional isolation. Okay. Now, examples of another example of sympatric speciation is behavioral isolation, and it is more related to the behavior of the organism. For example, there are small fish that live in salt water that have, with time, elaborated a certain mating behavior. This is also a kind of evolution. That mating behavior, how does it occur? When there is increased sunlight, the male will change its color and will develop a red underbelly. So there will be a red underbelly for this saltfish if that fish is a male. When sunlight falls on the surface of the body of the fish. And then the male will build a nest, okay? When this occurs, the male knows that this is the reproduction time, and it courts the female with a dance that triggers a complex set of movement between the two partners. So the fish starts doing movements like dancing, and this will trigger, as a response, a complex set. When the female sees this, it starts doing the same movements. If the female succeeds in doing those movements, those complex dancing movements, then it is a suitable partner. It belongs to the same species. But if it fails in any of the steps, see how rigorous they are, then it is not the proper mate, and it is avoided. And this is how reproductive isolation occurs, through behavioral isolation. You can do the same movements, then mating will occur. You cannot do the same movement, then mating will not occur. The same applies to the fireflies. Notice that the change occurs first in the male. In the male fireflies, they signal to females of the same kind by blinking lights on their tails in a particular pattern. Of course, if the female belongs to the same species, then it will respond and flashing back to the male to attract the male. So the female fish attracts the male by flashing back. If it does not flash back, then it is understandable that this female is not of the same species, and that's how they get isolated from each other. Of course, at the beginning, because you might be asking yourself, how of the different species? At the beginning, they were all one population, but there are variants that can flash and variants that cannot flash. So only those that can flash will attract each other, and those that do not flash, maybe they would disappear, or maybe they would have another mating behavior. But actually, at the end of the day, those that flash will not mate with those that do not flash, and there is some kind of reproductive isolation, but this time because of behavioral isolation, and we specifically mean the mating behavior. A fourth type of sympatric speciation that results from reproductive isolation, but not because of geographical barriers, is the temporal isolation. And temporal here means to time. Temporal is an adjective derived from time. So, for example, if we have a flowering plant species that has several populations, then some of those flowering plants will live in areas for a reason or another. They would be adapted more to sunny areas, and others will be adapted more to shaded areas. Of course, by the way, it might be in the same field. It might be in the same garden. If we have a roof here, for example, a roof of a if we have a building here above the flowers, then those flowers will be in the shade, whereas those ones will be in the sun. So they belong to the same species. Not only do they belong to the same species, but they might be in the same garden. But in that garden, sun rays, when the sun rises, sun rays usually reach until here. And here there is shade because of this part of the building, like a balcony or whatever that is above the flowers. So those that live in the warm areas will get mature more rapidly, and therefore they will form their pollen grains and their ovules more rapidly or sooner than the other flowers in the cooler areas. And therefore the pollen grains, the flowers will either self-pollinate themselves, or they will cross-pollinate the flowers that are in the neighborhood, but they will not pollinate the ones in the shade, because the ones in the shade are not ready to be pollinated. So this is a difference in timing because of different environments. And finally, we have number 5, reproductive isolation. This sounds weird. The title sounds weird. Why are we saying now reproductive isolation if all the previous factors resulted in reproductive isolation? Because here, this title is talking specifically about impossibility of reproduction because of barriers in the process of reproduction itself, in the process of mating itself. For example, we might have individuals belonging to the same species, but there are reproductive barriers, and those barriers might be pre-zygotic barriers, meaning before the formation of the zygote, and therefore they prevent mating, and post-zygotic barrier, meaning that mating occurred, but there are no fertile offspring. In pre-zygotic barriers, for example, it might be due to difference in the structure of the genitalia that prevents insemination, that prevents locating the sperms in the proper position, maybe because, for example, one animal is too big and the other animal is too small. The second pre-zygotic barrier is a difference in flower shape, because maybe the pollen grains of one population are more adapted to fall on a particular stigma that is flat, for example. So if the stigma is convex, the pollen grains would fall. So this is another pre-zygotic barrier. Again, the difference in size in number 3, but that is not related to the genitalia themselves, but related to the whole size of the animal. The post-zygotic barriers is what prevents a fertile offspring, meaning that there is fertilization, mating occurs, there is the production of an offspring, but it is infertile, like the example of the mule. And now we're going to talk about the patterns of evolution, the different shapes of evolution. So we have divergent evolution, and we have just talked about divergent evolution. When we have we have a population, an initial population, and then this notice that we haven't yet mentioned the word species. We have an initial population, and then this population loses part of it, and they become isolated from the rest of the species, and they become exposed to new selective pressures. Like, for example, if you have a population that lives at the bottom of a mountain, and then all of a sudden a small population lives at the top of the mountain. Why does this lead to divergent evolution? Because everyone, the original population and the small subpopulation that was separated from it are now in two different environments, and they are exposed to new selective pressures. So all the examples that we have mentioned in allopatric speciation and sympatric speciation are, of course, examples of divergent evolution. But we have another pattern of evolution that is convergent evolution. Here, in divergent evolution, we started by a common ancestor, and then all of a sudden, a subpopulation was separated from that common ancestor. So actually, they diverged from each other. So if we have population A, the original population, and then this is what remains from the original population, and that one, for example, is the one that got separated. In convergent evolution, we start by two different ancestors, by two different ancestral populations, but because they occupy the same environment, then they will be exposed to the same pressures, to the same selective pressures, and they have to show similar adaptation. For example, we have the shark and we have the whale. For a reason or another, the whale and the shark both live in watery environments. The shark is originally a fish and the whale is originally a mammal. But the whale is a mammal, for example, that has fins. So maybe this was a variant of mammals that has fins. At the beginning, it was a minority, and then that population of whales that found itself in the water found themselves very well adapted to water, and then they flourished in water until all the whales that are in water are survivors and flourish, whereas those that decided to live on land would perish. Whereas on the other hand, the shark is a fish. So although they belong to two different species of, they are both vertebrates, but they are two different species, yet because they live in the same environment and they are exposed to the same selective pressures of the water movements and so on, yet they developed, for example, similar characteristics like streamline appearance of both, for example, the presence of the fins. And by the way, the word developed a streamline is totally wrong. I'm sorry, I apologize, because they did not develop a streamline shape of the body, but those who had that streamline shape of the body were favored, and they were selected for. So when you talk evolution, don't say developed, because they did not develop fins. They did not develop a streamline shape of the body. They already have these things, and these things were favorable to the environment, so they were perpetuated, okay? And then we have a third example, which is parallel evolution. And in parallel evolution, it's true that we are talking about two species that diverged from a common ancestor, but they are two related species. And therefore, they make similar adaptations even after they diverge. So in parallel evolution, we start by two different species, but the two species are related to each other. They arise from a common ancestor, and that's why it is a type of divergent evolution. But after having diverged from the common ancestor, they follow similar evolutionary adaptations. And so essentially, they diverged from a common ancestor, but later on, they showed parallel evolution. And the classic example is the example that we mentioned at the beginning of the marsupial mammals, like the Tasmanian wolf and the placental mammals. The marsupial mammals are found exclusively in Australia, And the placental mammals are found everywhere, including North America, and for example, the gray wolf, and they have striking similarities because at the beginning, when all the continents were the Pangea, or when all the continents are continuous with each other, animals could freely move over land. So the marsupial mammals were established in Australia because it was the most favorable environment for them. Whereas, for example, the placental mammals stayed in the ones that stayed in North America found the environment favorable. So they share a common ancestor, but even after they diverged, so at the beginning they diverged, maybe because of geographic isolation, but at the end of the day, after having diverged, they had the same environmental pressures and therefore they had the same adaptations. Okay. The coevolution, on the other hand, in coevolution, we still have two interacting species, meaning that the two species affect each other, and there is a reciprocal set of adaptations, meaning that they will affect each other. The two species, A and B, will affect each other, and this is what we call coevolution. Of course, the best example for coevolution is the predator-prey relationship. All predator-prey relationships will be considered into coevolution because, as you saw, for example, the hare is being eaten by the lynx, and then the lynx is being affected by the diminution in the number of the hare. So here we have two interactive species, but these two interactive species are not interrelated, but they are interactive. Notice that here we are not explaining how species develop. We are not explaining speciation. We are explaining patterns of evolution. So co-evolution, as you know, the word co means at the same time. Co-evolution, or not at the same time actually, but co-evolution just simply means like this mutual interaction between the two. Here, in that context, I mean, so predator-prey relationships, of course, are an example of co-evolution because the hare affects the lynx and the lynx affects the hare. And then we have a famous example that is the relationship between the monarch butterfly and the milkweed plant. The milkweed plant is a plant that contains poisons. So the milkweed plant is a poisonous plant. Hello again. I have to apologize because I've just checked the pronunciation of the butterfly. It's monarch. Okay, so this CH is pronounced K. So we say the monarch butterfly. So how is the relationship between the monarch butterfly and the milkweed plant a kind of co-evolution? The milkweed plant is a poisonous plant, as we said before, and the monarch butterfly lays its eggs on this poisonous plant. So when the larvae hatch, all what they feed upon is the milkweed plant, and therefore the larvae themselves become poisonous. And when the larvae grow up, they become the butterfly, the monarch butterfly, the adult butterfly, and the adult butterfly is therefore poisonous. And because the adult butterfly is poisonous, and just like any other poisonous insect, it has very bright and conspicuous colors. And usually these bright conspicuous colors are protective for the butterfly, and meanwhile protective for the milkweed because they tell the predators that we are poisonous. So the milkweed plant is beneficial to the monarch butterfly because the eggs of the monarch butterfly are being led there, and the larvae and the insect feed on the sap of the plant and absorb the poisonous substances. And meanwhile, the butterfly, being colorful, being brightly colored, will deter the predators. So there is like mutual exchange of benefits between the two. Now, the fifth type of radiation is, or the fifth type of evolution, the fifth pattern is adaptive radiation. Adaptive radiation is a bit similar to divergent evolution because the species develop from a common ancestor, and every species will be lodged in a particular niche, as we said before, where it will be best adapted. So it is similar to divergent evolution, but this time we have several divergences in several niches or in several comfortable bubbles or comfortable environment. So, for example, all the 14 species that Darwin, species of finches, birds that Darwin described on the Galapagos Island, they all diverged from one ancestral species. Maybe when they sequenced their DNA, they determined the presence of many similarities between them that indicated a common ancestor, but actually there are currently 6 species of ground finch, 6 tree finches, 1 warbler finch, and 1 bud eater. Of course, these represent 4 different species that all originated from the common ancestor. They started as populations that split from the original population, and every population was lodged or inhabited a particular niche. And now we're going to talk about the theories of evolution. The first theory of evolution is the theory of gradualism. And this theory says that, or postulates that, the organisms descend from a common ancestor gradually, that they descend from a common ancestor over a long period of time, and that they descend from a common ancestor either in a linear fashion or in a branching fashion, meaning that, for example, if we have an ancestor A, there could be divergent evolution to produce ancestor B. This is what we call the branching fashion. And I'll not be explaining now the linear and the branching fashion until we discuss what we call the phylogenetic trees. Maybe if they're not on the slides, we're going to discuss the phylogenetic trees that determine, that show this branching of new or the branching evolution. If it is on the slides, I'll be explaining it. If not, I'll be explaining it through the homework assignments. So this theory of gradualism, up till now, there is almost all of the postulates are correct, because evolution is a gradual process. Evolution occurs over a long period of time. Evolution usually occurs in a branching fashion, and the big changes that occur in evolution, as we said before, are due to accumulation of small changes. And at every stage, for example, if we have here another species that diverges, then, for example, A is the ancestral species, so it is the oldest species. C is the most recent one. But actually, if you have fossils for all these organisms, you can use those fossils, as we said before, for determining the age of you can use radioactive material to determine the age of the fossil. And in order for this theory of gradualism to be a correct theory, then you should have fossils for every stage of these. You should have fossils for the ancestor. You should have fossils for species B, if you assume that there are that there is a species B, there should be fossils for C, and there should be no missing links in between. But of course, the fossil record is at odds with this theory because the scientists rarely find transitional forms in between the species. We're going to stop here, and another part will be sent on Google Classroom. Thank you so much, and see you in the fourth and last part of evolution.